Hello everyone, welcome back. So welcome to Akshit's second talk. So he'll today walk through a couple of problems detailing some of the ideas that he told us yesterday. Akshit.
Hi everyone in the room and everyone on Zoom. If you missed my lecture yesterday, okay. All right, yes, please interrupt whenever something is unclear or when you need to ask a question. If you missed my lecture yesterday, I'm going to emphasize that's not going to be a problem. I'm going to talk about a slightly different set of problems today. And I'll take you through the prerequisites that you need for it in the lecture itself. Just to sort of start off what we're going to be doing today. Ecology is essentially the study of organisms and how they interact with their environment. In a very crude way of speaking, there are organisms, they can modify the environment around them, and there's a feedback loop where the modified environment thereby impacts the growth and survival of these organisms. And the result of all of these interactions between the organisms and their environment are various phenomena that we can observe around us. And here's 3 examples. This is by no means an exhaustive list, but one phenomenon that we sort of robustly observe in almost all ecosystems is their stunning diversity. That is, wherever we sort of go and look, whether it's in the world of plants and animals, or whether it's in the world of microbes that are in us and all around us, we see hundreds of thousands of species being able to coexist. And so an important question that sort of is there in ecology is, how can we predict and put good bounds on how many species should be able to coexist given some details about the environment? Another phenomenon that we observe quite robustly in most ecosystems is the fact that they exhibit multi-stability. That is, they have a small number of multiple stable states. In some of these stable states, you get different organisms being able to survive. The kind of multiple stable states that we're going to be talking about today are slightly less well understood, but nevertheless as common, which are multiple states that have the same set of organisms surviving, but at different levels. So that's sort of the second thing that we're going to be talking about today. The third thing, which I spent some time talking about yesterday, are sort of dynamics and in particular correlated dynamics across different levels. And so I'm not going to be talking about that today, and I'm not going to be talking about various other phenomena that can happen in ecosystems. And sort of the 2 questions that we're going to address are going to be by means of 2 different models, with a time thread that they're both going to be modeling the environment in terms of the resources that microbes can consume. In the first sort of story, in the first model, we're going to be using a dynamical systems and asking whether we can put reasonable bounds on how many species can coexist reasonably in any ecosystem. And in the second story, we're going to use ideas from game theory, in particular the stable marriage problem, to be able to map something to microbial, map that problem to microbial ecosystems and describe how we can get multiple stable states and transitions within them. So let me start sort of with story 1, which is about diversity. And I thought it's sort of good to start off with a sort of illustrative example of why people talk about diversity and why people sort of worry about it. And that has to come from this idea called the competitive exclusion principle, which says that the number of species in any environment is strictly bounded from above by the number of resources in that environment. And there are sorts of many calculations that you can do to show this. I'll show you one such calculation. Imagine sort of a simple experimental system that you can build on the bench top. This is called a chemostat, where what you essentially have is that you're flowing in some nutrients. There's some inflow of nutrients at some rate phi 0, and we're going to imagine for simplicity that there's only one nutrient, one carbon source, if you will, called C0, and we put in one bacterial species, which we call B1. The abundance of this species we'll track is B1, and this species can consume this particular carbon source. Why is this thing called a chemostat? Because, of course, if we keep just flowing in the resource, it will keep accumulating, the bacterium will keep using it, and will keep growing uncontrollably at an exponential rate. So what you can also do is that you can create a sort of outflow in the system by means of dilution at a certain rate delta. And what you're diluting is essentially both C0 and B1 at a certain rate. And in this system, we can write down the dynamics of this system and study its steady state, sort of see why this competitive exclusion principle must hold. So the way that we will write down the dynamics is that we'll write down the dynamics of our resource C0, and we'll write down the dynamics of our bacterial species B1. And let's start with writing the dynamics of this bacterial species B1 first. We're going to assume that it consumes the resource at some rate, lambda 1, and its growth rate is then proportional to lambda 1 C0 B1. That is to say, there's some per capita, per unit concentration growth rate that it has, which we call lambda 1. This could be in principle a function of how fast it's able to accumulate resources and so on and so forth. And the more the resources, the more it grows linearly, and the more the population, the more it grows. And of course, this species is diluted at a rate delta, right? And so the dynamics of the species are quite simple. There's just a growth term and there's just a dilution term. What about the dynamics of the carbon source itself? Well, there are three things happening to this carbon source. It's flowing in at a certain rate phi 0. It's being consumed by the bacterium at a certain rate, and then it's also diluting away, right? And we're going to make the following important assumption, which is that when this bacterium is actually using the carbon source, it can't actually completely convert the energy in the carbon source. There's always going to be some inefficiency. And so we're going to imagine a yield term Y, which is going to tell us how efficient this particular species is at consuming the resource. And so the way that's going to reflect in the dynamics of our system is going to be that the resource is not going to be depleted at this rate lambda 1 C0 B1, but at some rate divided by Y. And this Y is strictly speaking less than 1. And again, a dilution term, which is proportional to C0, right? If we want to calculate the steady state of this system, and if you want to know what the bacterial density, what the bacterial abundance is at steady state, we only need to sort of solve these two equations and put them to 0. If you put this equation to 0, that's correct. That's correct. Yeah. So the imagination is that there's some sort of hole here in the system, and that sort of things are flowing out. Right. And so the rates of these two are in this model the same. In principle, they could be different. If you solve this equation, if you solve this pair of equations for steady state, you will find that when you solve the equation for B1, you essentially learn that the concentration of this carbon source at steady state, which I'm going to represent by this asterisk here, is completely independent of the bacterial concentration itself, and it only depends on the dilution rate and the growth rate of this particular species, right? I'll get to solving this equation in a bit, but I sort of for the purposes of illustrating the competitive exclusion principle, this will be enough. If you sort of observe what this equation is telling us, what it's telling us is that the steady state concentration of this resource is dependent essentially on lambda 1, that is the growth rate of this particular species. And so the larger and larger this lambda is, the lower and lower the concentration of this resource will be at steady state, right? So now imagine a scenario where instead of just species B1, you have in principle two different species, B1 and B2. If you solve with sort of dynamics that look sort of exactly like this, except with the lambda 2 instead.
This is steady state for dB1 by dt, but there's dC0 by dt, which so you're going to set the source in some ways as dC0 by dt is also 0. You're just not.
That's correct. Yeah. So we put both of these to 0, except that by solving this equation, by putting this to 0 itself, we learn sort of that the steady state concentration of C0 is completely sort of set by that.
I see. And then the source is whatever it is that you get by solving the first.
That's correct. So in fact, the steady state concentration of this resource is completely independent of the supply rate.
So if the supply rate is arbitrary, you won't reach a constant C0, no?
Correct.
So it's not independent. You mean that you can, if you solve for dB1 by dt, then you set C0.
Yes. Yeah. Correct. So yes. So the assumption that you're making when you're solving this equation is that C0 has already reached steady state. Correct. Okay. Yeah. So that's a requirement. Right. So if you make phi naught infinite, of course, then this thing will not hold. Right. So the argument basically goes that if you have two different species, they'll sort of drive down the concentration of this carbon source. The species which has a higher lambda will sort of drive the carbon concentration lower, rendering the species with sort of the lower lambda with a negative growth rate essentially, and that species will go extinct. And this sort of is a simple illustration of how two different species with two different lambdas, which is in general going to be true, cannot survive together in this environment on just one carbon source, right?
Sorry, I have a question.
Yes.
I guess I'm confused. Your definition of competitive exclusion, is that inequality?
That is an inequality, correct. So in principle,
what you've explained so far, right?
Right, yeah. So this is just an illustration for one resource. In principle now, if you have multiple resources, you could have one species in principle surviving on each of them. And on some resources, you could have it so that species cannot get a positive growth rate on them at all.
So, but how do I get the inequality that the number of species is less than or equal to the number of resources?
Right.
That's something about the dimension of your dynamical system.
Yeah. So certainly, if you try to put more species than resources, you wouldn't be able to simultaneously satisfy all of the equations that you would write for them. But certainly, if you put less species than the number of resources, you could get them all to survive. That's sort of what the argument is for the inequality.
Is it possible that I have a large number of species, but many of them have so few members, so that the number of species, I try to violate that inequality, but I try to remain close to your equilibrium by setting a lot of them to epsilon?
Let me see. So you're trying to say if I set their steady state abundances to some really small number,
that's right.
Could I get an arbitrarily large number of species to survive?
That's right.
I don't think that that's going to be true. The reason being that the calculation for the steady state concentration of this resource, the steady state concentration of this resource is going to be some number, right? Finally, and that itself is independent of what the steady state abundance of your species is. So, okay. So you could set it to an arbitrarily small or large number in that.
I mean, the assumption that's going in here is that of complete homogeneity, right? That all the species dilute at the same rate, and
that's correct. That all the deltas are the same.
And so on, right? If you violated that and allowed, then you would have
correct. Correct. And in fact, sometimes when practically people actually build these kind of chemostats, what they find is that lots of species can actually survive by sticking to the walls. And so essentially they get a delta of 0.
Akshit's question.
So the question was that in principle, this calculation, there was a central assumption that the deltas were the same for all species and all resources. And the answer was that yes, that's true. That's required. Yes.
Hello.
Akshit, can you hear me?
Yes.
Yeah, so when you say survival, you mean survival at infinite time. Like it could be that, dynamics is very slow and many things are surviving.
It could be that you could construct models where there were very long-lived transients. And in fact, people have constructed certain ecological models which have even chaotic dynamics where all of the species can coexist. But that's sort of not the example that we are taking here. But you're right. We're only looking at sort of the long-term limit.
Thanks.
Hello. I was wondering whether this model may be too simplified in that in actual systems, it could be that the bacteria B1, after consuming the resource C0, produces another resource.
You stole my punchline.
Oh, okay.
Right.
Yeah, because
absolutely,
you've taken the resource, but then you generate something else.
Absolutely. Yeah, correct. Did you have another question to follow up on that?
No. I
okay, great.
That could evade this, I would imagine.
That's correct. And so essentially what we're going to do in the next sort of 20 or so minutes is to study what happens as a result of this yield not being perfectly one. What can happen is that this species, while consuming the consuming resource C0, can actually produce a whole set of byproducts as a result. And it's these byproducts that can serve as new carbon sources for new species, allowing all of them to survive. So just by studying the dynamics of all of the different byproducts, we can recalculate a sort of new upper bound on how many species can exist in this system. Any more questions about this? Yes.
Actually, can I just? Okay, so maybe you explain this, but how do you go from here to the number of species? I can understand how you might say from this whether a species can go extinct or whether it can survive.
Correct.
But how do you go from a multiple number of species from there?
Yeah, no, this is a good point. The question, just to repeat for those who didn't hear on Zoom, is this is a constructed example for one resource and multiple species. But what about a more general example of multiple species and multiple resources? There are various ways to construct it. For purposes of time, I'm going to stick with the special case of this, which is going to be an environment where there's just one resource that's provided. That's sort of the egregious example of this. We're going to see that currently, while the limit might be one species, we can grow many more. But I can chat with you separately to show you how we get those sort of more general models. Those are just not my work, and they've been done before, so I'm not delving into it.
Sorry, but why is it complicated? We just multiply this equation by n. So you'll have n resources and n species, right?
Right. The complication comes in only because in certain models, what people assume, the sort of key assumption that we had here was that this species is only capable of consuming this one resource. If you have multiple resources, then in principle, all species could be consuming.
It's a matrix eigenvalue problem.
That's correct. That's correct.
The number of eigenvalues is the size of the matrix.
Precisely. Yes. Okay. Just because it's going to be useful later, let's also solve this equation to sort of get what the bacterial abundance at steady state is going to be. And if we solve sort of this equation, I won't solve it fully, but we end up getting something that looks like this. And for that, we make the assumption that C0 has, equilibrates quite fast, and so we can put the steady state abundance of C0 in these equations and just solve at dc dt equals 0. right? And so there are a few interesting things to note about the bacterial abundance at steady state. The first is, sorry, was there a question? Okay, sorry. The first is that, of course, the higher the dilution rate, the lower and lower this bacterial abundance gets. And of course, the higher the yield of this bacterium is, the higher and higher its abundance gets. But there's sort of, the other dependence is on this sort of term in the parentheses, which you can think of as a sort of effective flux of the resource that it's getting, which is just basically the flux, the inflow of the resource minus delta times the steady state abundance of the resource, right? And so that sort of adjusted flux is something that we're going to come to. Okay, so like Rajesh was saying, it's not necessarily true that this is somewhat a simplified picture, and that it's not necessarily true that you're just going to have this one species consume this one resource. What is in principle going to happen is that this particular species consumes this resource and sort of eats up a fraction y of it, and a fraction 1 minus y of it, it's able to secrete out as various byproducts, and we can imagine there are beta byproducts in principle, right? And so these byproducts can then now serve as new carbon sources for new species that can now survive, right? And so that's sort of going to be the central idea that we're going to explore in sort of this part of the talk. Just in terms of the parameters of the model, we're going to assume that there are beta byproducts every time a species consumes a resource. Just for simplicity, we're going to assume this beta is universal. In principle, the calculations that we're going to do are going to be with beta equals 2. But in principle, they could be with anything. The second thing that we're going to assume is, just for simplicity, we're going to assume that y, our yield y, is some universal yield which can be thought of as 1 minus alpha. So we're just going to think of, instead of y, we're just going to think of 1 minus alpha. And so the fraction alpha of whatever resource is consumed is going to be secreted as byproducts, right? And so now, without loss of generality, we can just write these resources as C1 and C2. And we can write down new dynamical equations for these resources and for any species consuming these resources. So let me do that for one of these resources. So for dC1 by dt, it's again given now by the inflow of this resource, and the inflow doesn't come from the top, it comes from the production of this resource. And again, it's sort of consumed by, we're just going to assume that it's consumed by some species B2. And then, that species could in principle also produce more byproducts, right? And so there's going to be 3 terms here. One is going to correspond to the production, one is going to correspond to the consumption, and one is going to correspond to dilution, right? And so for production, we basically just have the rate of consumption of C1 modified by the fact that a fraction alpha of it is what is produced as byproducts, and that's split into beta byproducts in principle. And just to simplify our calculation, we're going to assume that all byproducts are sort of equally split. And so that's the fraction that goes into the production of C1. The consumption of C1 is going to be done by this species B2, and that's going to be with some lambda 2 C1 B2 by its own y. And again, we're assuming that all of the yields are some sort of universal constant, though in principle they could be different. And again, we're going to assume some sort of dilution, right? And was there a question?
Yeah, sorry, just an algebraic question. I thought the amount consumed is 1 by 1 minus alpha, right? I thought you seem to be assuming that it's 1 plus alpha. Are you setting 1 by y equal to 1 plus alpha? Or are you?
Sorry, I'm setting y to be 1 minus alpha.
I see. So then the amount of the resource that's consumed is 1 by 1 minus alpha, right? That's dC0 by dt. I mean, the way you defined the equation originally, because you had 1 over y, so it's 1 by 1 minus alpha.
So I'm assuming that you're sort of consuming a certain flux of this resource.
Right.
A fraction 1 minus alpha is going into the bacterial biomass, and the remaining fraction alpha that flux is going into byproduct.
I see.
Yeah. So you could, So you could imagine, instead of writing this here, you can you can imagine writing 1 over y here.
I see. Okay.
Right.
Okay.
Yeah, and that's just a matter of convention. Okay. Any more questions about this equation? Right. So this is just the production by the species at top, the consumption, and the dilution. And again,
I just ask a question.
Yes.
So in a realistic situation, I would expect that the species themselves may be consumed by other species, right?
Okay. Right. So definitely, if you're thinking about sort of the macro world where there are plants and animals, that's certainly true that the species are going to be consuming other species. We're sort of imagining a world where all of our players are different microbes. And so in microbes, it's generally not true that they eat each other. What they eat is sort of chemicals that are secreted by each other.
I see. Okay. Thank you.
Right. And so the equation for sort of the species dynamics is just going to be very similar to the species dynamics that we had before, which is that there's some lambda C1B2 and there's some dilution, right? And so this equation is always going to give us the steady state concentration of the resource that species is consuming. Right. And so we're sort of always going to be interested in calculating then the concentration of the bacterial biomass by sort of using this steady state concentration and sort of putting it here, right? And so if we do that, you can sort of put that in here and go solve the algebra. I won't do it here for sake of time. What you sort of get again is that you can write down the steady state concentration of B2 as again some phi effective y by delta, very much like what we had for B1, except now the phi effective looks slightly different. The phi effective looks as follows. Right. Okay. And so basically the idea here is that you can always think of the bacterial biomass as being, of course, affected by dilution and the yields, but sort of always going to be proportional to the flux of the resource that it's consuming that's sort of coming in. The sort of lower and lower this flux gets, the lower and lower the concentration of this bacterium at sort of steady state gets. And of course, you're going to get species being able to survive until a point that this phi effective becomes 0. Right? So in principle, what you can do is that it's sort of, as you go down these layers, you get the same steady state equations, except the bacterial biomass is always affected by this phi effective. And if you're at layer L, you can solve always the fact that the phi effective always looks like this, right? And so just to be clear about what I mean by L, species C0 is at L equals 0. It's sort of the species at the top. Species B2 and whatever is consuming this resource are at level 1. Below them will be level 2, and so on and so forth, right? And so at each level, you have beta to the L species that can survive. And this is going to be true until your phi effective becomes 0. Are there any questions about this?
Yeah, just a silly confusion. I think if you put alpha equal to 0, why is the concentration, the flux, there's still a non-zero flux and I thought, shouldn't alpha equal to 0 take you back to the original situation?
So when alpha equals 0, you don't have anything being produced, right?
Exactly.
So when alpha equals 0, this term goes to 0.
Right. But there's a non-zero term in the second.
Right. And so basically this term is some sort of negative term. Right. And so what this is saying is that your bacterial biomass at steady state will be some negative, where not well defined.
Regime,
right. Correct. It's just not a physical regime. There was one more question on Zoom. Oh, sorry. Here.
I had a question. Sorry, maybe you already mentioned this, but so you are sort of assuming, I guess, some sort of adiabatic approximation or something like that, where you're assuming the C1 reaches steady state fastest, right?
That's correct.
But the thing is, so in which parameter regime is this approximation valid? Because on the other hand, you can, perhaps even at least numerically solve the actual differential equation. And can you say in which parameter regime this approximation is valid? The fact that C1 is reaching steady state faster, and then you just use the solution of C1 and plug it back, right? What is the regime of validity?
Right. Yeah. So that's a good question. So it's typically when sort of your dilution rate delta is large. That is, it's larger than the growth rate, the sort of the instantaneous growth rate of your species. It's important in this calculation because to solve the bacterial abundance in steady state, you need to put in what C0 will be. And so what we do here to do this calculation is that we calculate, we sort of put C0 at steady state.
All the dBi by dt equal to 0, you're saying that does not have a unique solution?
If you simultaneously try to solve sort of all of these equations together.
No, you don't need to solve the dC0, right? You just need to solve dB1 by dt because there are sources.
Right.
So you just solve dB1 by dt equal to 0 and dB2 by dt equal to 0.
Right.
Simultaneously. Doesn't that have a unique solution?
Yeah, so my question is actually related.
I think you have 4 unknowns then, right? Huh? You have 4 unknowns and 2 equations.
It seems that you do, right? Because the first one doesn't depend on B2 at all. So you get C0 as B1 by, you get C0 as delta by lambda 1 from the first one. Unambiguous.
From which one? From
this
here?
One, the one below that.
Yes, but here we've already, by solving this equation, we already get C0 at steady state, right?
No, I'm just setting all the dBIs by dt to 0 simultaneously. I'm just trying to ask what is the one simultaneous steady state? I don't care.
Correct. Yes.
And I think you get it unambiguously. You don't need to assume it came first or second or anything, right?
If you just solve the dt equations, you get, yes, correct. So what you get is you get all of the resource concentrations at steady state simultaneously.
Right.
But now if you want to solve for the bacterial abundances at steady state, you'll need to solve this set of equations. Right? Because you don't get the bacterial abundances at steady state from solving
all
the dBdts.
No, but I guess you can solve for the time dynamics of everything, right? Like at least numerically for C0, B1 and so on. And then you will presumably get some steady state. Let's say you get a steady state value of B1 for very long times. Right. How does that compare to this approximated version? Like,
it works quite well, and I'll show you a few simulations in a bit.
Okay, thanks.
Yeah. Okay.
But in principle, one should be able to identify all the fixed points and their domain of attraction, right? What initial condition up and at which fixed point? Isn't that possible?
Yeah, so in this case, you have one fixed point that's globally stable. There are no multiple fixed points in these equations.
So there is only one equilibrium result.
That's correct.
And so you'll always reach there. Isn't that true?
Yeah, true.
Okay.
Can I just make a comment here?
Yes, please.
There is one solution with if you assume that B1 is not 0, then you get a solution that C0 star is delta by lambda.
Yes.
Right.
Right.
But there is always another solution in which B1 is equal to 0. That is, the species has gone extinct.
Correct.
So C naught is not then fixed by this equation, but it might be fixed by the second equation, by the second bacterium.
But then you don't have the second bacterium at all anymore once B1 is 0, right?
No, I mean, I was referring to your earlier example, not the one that you are now having.
Okay, I see. Yes.
I was referring to the equations earlier where you were illustrating the competitive exclusion principle.
Yes.
I just want to make the comment that there are actually multiple solutions if you allow for the bees to become extinct. And that is the point, that either you will have a solution with B1 equal to 0, or you will have a solution with B2 equal to 0. Both cannot simultaneously be non-zero. That's the conclusion from the first set of equations. And C naught, of course, will depend upon which one is the larger lambda of the two. That is what will determine C naught.
Correct. Yeah, and in fact that's true. Even here, my statement of a single equilibrium is true for one with all of the species and resource concentrations being non-zero and positive.
Yes.
Ashok's previous question is whether there's a domain of attraction. So even though the fixed point might be one with the concentration is,
right.
The species is 0, but it might have a domain of attraction.
Right.
Right.
So in this case, if you start from the trivial fixed point, which is where all of the concentrations of the species are 0, then you'll stay there, of course. But if you start off with even a small amount of the species,
so then you're saying that's unstable. That fixed point.
The fixed point 0.
Yeah. That if you start off even with in the vicinity of that fixed point, it will be driven to the other fixed point.
Yes. Yeah. Correct. So you can start off with a really small amount of each of these species and be taken to these equilibria.
So do we have a phase portrait? It would be easy to, at least even in maybe, 4-dimensional, not necessarily in all 4 variables, but is there a way to visualize that? I think that's probably the confusion here, because even it might be unstable in some directions, stable in other directions. You want to know what is the nature of that unstable fixed point? Is it a saddle or a...
Yeah, no. So basically, there's essentially, if you draw this phase portrait along any of the axes, let's say you draw it along the axis of B1, what you'll find is that basically there's 0, which is the trivial fixed point, and then there's this other fixed point that's given by this equation, and basically wherever else you start from, you're sort of attracted.
That axis is an unstable direction, but there might be in the bigger phase space there might be
where you have B2 and sort of right.
So that's
the
question.
So my understanding is that again in that sort of bigger space you just have 2 fixed points. One is 0, 0, 0, 0, and then one is sort of this fixed point that's given by C0 star, B1 star, C1 star, and B2 star. And there's and anywhere you start, which is not the fixed point that's 0, you sort of stably go towards this fixed point.
Can you do a linear stability analysis about the trivial fixed point and find out the number of stable and unstable?
Yes, you could. So you could sort of perturb each of these steady states and see that. I'm not going to do that today just because I want to do a different calculation. I want to calculate how many species can coexist, but certainly you can do that.
What is
Yes.
What is the most general problem here? You see, you're doing C0, B1, and then adding something else. What is the general problem actually, and the general setup? And to be able to analyze even qualitatively actually.
Right. Okay. So the general setup, that's a good question, and that's what I'm getting to now, is that you can basically imagine this process of species consuming a resource and then producing byproducts and new species consuming this resource, and this process going on for multiple layers until a point where species abundance, and so as you go down the layers, because the effective concentrations of each of the resources are going down, the effective steady state abundances of these species are going down. This continues until a point, and that point is when the effective flux of the resource that's coming in at some layer cannot fight dilution enough. And that's the limit of how many layers this ecosystem can have. And so if we can calculate what that bottommost layer is, we can then calculate in principle what the maximum number of species that can coexist in this system are. And so what we're trying to calculate is what that maximum number of species is as a function of the various parameters in the system. And we're going to see that it depends only on the dilution rate, really.
Yes. Just, to understand these 6-point questions, if I understood it right, the statement was that if you have a fixed point, which is just made of, like a bacteria with a smaller fitness, you could perturb it by adding a bacteria with a higher fitness, and then it will drive it, and then it will drive the
other one to extinction.
It will drive the lower fitness bacteria to extinction, and then it will go to the fixed point where you have a higher fitness bacteria.
Correct.
And in principle, the only stable fixed point is with the bacteria with the highest fitness.
That's correct. Yeah.
Okay.
Yes.
For each resource, in some sense.
That's correct. Yes.
So that was a competitive exclusion model. Right?
That's correct.
So now when you add, these layers of where you have byproducts and so on. So one question about the fixed points would be that, can you have, let's say, 2 bacteria, which if they were not, eating each other's resources and so on, would have been less fit, that there would have been a higher fitness bacteria which you can put, and then you could drive it, to some other fixed point. Okay, could they become, by cooperation, in some sense, their total fitness is better than what it would have been? Is that the kind of thing that would happen in this model?
Let me see if I understand this correctly. So you're saying that right now the species are sort of splitting turf and they're only eating the byproducts of what is being produced. Could they do better by not just doing this, but sort of by eating multiple things at the same time? In principle, they could. In this simple model, at least they don't. But you could imagine more complicated models where one bacterium is not just eating one resource, but in fact multiple resources, and now there's competition that's occurring across levels, and that's a more complicated model that I haven't analyzed.
Okay, so the statement is that in this model it is still true.
Yes.
That if I take at any level a bacteria,
yes,
and if I introduce a bacteria with a higher fitness than that bacteria, which
on the same resource,
on the same byproduct
yes,
or whatever at that level,
correct,
it will drive this to extinction, and you'll be driven to a fixed point where there's the bacteria.
Correct, where basically everything will remain the same. You'll just replace the lambda of this new bacterium instead of the old bacterium.
So there's still some kind of a competitive exclusion, but it's now on the byproducts.
Yeah, correct. Yeah, so on every resource, there's sort of one resource competitive exclusion that I defined holds. That is, now you can imagine a picture where you're sort of throwing in multiple bacterial species into the system sort of one at a time, and they all use one of these resources at random. And whatever that resource happens to be, if it's currently free, if it's not being occupied by anyone else, then it just goes and sort of settles there, uses that resource, and then you get the fixed point concentration that is defined by these equations. And if it actually is potentially competing with another resource, then just knowing the lambdas of both of these species will tell you which of them will win and which of them will die. And just whichever has a higher lambda will win and replace the other species. So there's competitive exclusion occurring individually on all of these different resources. And again, the number of byproducts that you get plus the original resource is still the maximum number of species that you can get.
Question?
Yes.
So can I try to summarize what you just said in a way that I understand?
Yes.
Which is the connectivity from layer 1 to layer 2 or layer n to layer n is in principle decided by this principle of competitive exclusion.
Yes.
So it gives you a principal way to say what the number of connections or to be from layer to layer, and how to decide which species will be collected?
Yes.
Sorry, I didn't understand that answer.
Yeah, could you please repeat the question?
Can you repeat the question?
Yeah, okay. So I don't know if I'll be able to repeat it exactly, but the point here is that there's some sort of mapping from layer to layer of basically how many new connections or how many new resources you create, and just by knowing how the layer to layer mapping occurs, you can sort of figure out the entire structure of the system.
See, but are you? I mean, in this question, aren't you assuming that the bacteria as a second layer can only eat the byproducts of the one in the first layer?
That's correct.
In this model, it's not true that all the bacteria can consume all resources, right?
That's correct. Like I was saying to a previous question before, we make a simplifying assumption here, which is that we throw in many bacteria sort of one at a time, but each of them can only consume one resource.
But is that a realistic assumption? In that, is it true that bacteria can't substitute one resource for another?
It is true that bacteria usually can substitute one resource for another. Yes.
So then the more general model would be to have some matrix for all the resources and all the byproducts.
Correct. And so there's slight differences here. The sort of scenario where this would apply would be sort of one that I'm going to use in a bit later also, which is that there are some bacteria which, if you provide many resources, they consume all of them. They're capable of consuming all of them, and they consume them simultaneously. And then there are many bacteria which, if you provide again this set of resources, they are capable of consuming all of them, but they use them only one at a time, based on some internal regulatory hierarchy that they have. So if you present a bacterium with some really good sugar like glucose, they'll prefer to use it before they use anything else.
I see. But this other model would allow these more general phenomena that Lotka had, right? Where you could cooperate. One bacteria could
that's certainly true.
And become fitter.
That's certainly true. And sort of we sort of study these models as well. This is a sort of simpler, easier picture to understand than that more complicated general picture. Yes, please.
You are trying to explain to us the work you have done by some very simple, but can you tell me what exactly you did? I mean, what are the equations you're really working with? And they cannot be so simple as this.
They are these.
You should work with, let's say, N resources and M species of bacteria, with all the complications of, the terms that you have to put in, and perhaps even non-linearities, which you have not discussed at all. So that is the sort of problem that one should look at, and then look at the phase portrait and fixed points and qualitative physics of, I'm sorry, not physics, but the qualitative implications of that type of thing. Is it a stupid question? Tell me.
Yeah, no. I think it's fair that in general, one should try and understand what happens with N species and N resources with all of their complications. Nevertheless, I think it's sort of illustrative and good to show sort of, what happens in some simplifying cases, so that sort of we can build up more general
But
things.
what did you actually do? I mean, in your paper, in your work?
We solved these equations yes.
Only these equations?
Yes. I mean, up unto a certain number of layers, and what you find is that these sort of four sets of equations can.
So how many layers did you really take?
Right. So the number of layers is what I'm going to get to now. Right. So So the question was one of how many layers can we get, and the number of layers that we can get is actually a good question. So let's tackle that. So in principle, you can have a bacterium producing a set of resources, a set of byproducts, have other bacteria using it, producing a different set of byproducts, and so on and so forth. This sort of process can go on until a layer L max, right? And at L max, what happens is that phi effective goes to 0. That is, you can have no more bacteria survive. And so we can actually calculate what that L max is by knowing what our phi effective is. And basically, when phi effective goes to 0, what you get is an expression for L max, which is right. And basically, this tells us, given the dilution rate, given the initial flow rate of the resource at the top, and given these parameters alpha and beta, which tell you the yield and the number of byproducts, how many layers in principle you can have a bacteria. And remember, what we want to be able to do is to calculate how many species can now coexist in this system. And so we can note the following fact, which is that basically you're going to get beta to the power L species surviving on every layer, right? So you get one species at layer 0, two species, beta species at layer 1, beta square species at layer 2, and so on and so forth. And so the number of and so in principle, the number of species can survive that can survive in this ecosystem can be calculated by just adding up all of these different species at different layers. And that's just a simple geometric series. Sort of n max, the number of species that can survive, is just, beta to the 0 plus beta to the 1 plus beta square, all the way up to beta to the L max. At the last layer, you can get no more species than this. And this is sort of roughly beta to the L max plus 1 by beta minus 1. And we know what L max is from knowing what phi effective is. We don't need to solve all of the equations. And so we learn that basically N max in principle, if we substitute it here, scales as a power law that depends on delta. So just to write out the more general form of it, N max sort of approximately scales as e to the log beta log phi 0 plus log lambda minus 2 log delta. And so this really gives us this upper bound that we were looking for in the beginning, which is how many species can survive. And so in principle, another way to sort of write out this is to notice that it depends on delta. And you can sort of take that dependence out and note that it scales as a power law, which depends on delta with the following exponent. And so now what we basically learn is that the number of species can in principle be controlled by tuning the dilution rate of the system. The lower and lower the dilution rate, the more and more species you can get. Right. And so this sort of power law is one that we found that sort of tells us that this number of species can be quite large, even when you supply the ecosystem with one resource. And now you can imagine generalizations of this version where there are in principle multiple resources that you provide. Then you can sort of, again, use the same tricks to get what the number of species would be. And there's a whole bunch of different things you can do with this model. For instance, to compare it with data. One of them is to calculate actually the abundance distributions of all of the species that are going to exist in the system, and that scales according to a different power law, which I won't derive today because I want to get to the second story. I'll just share a few slides to show you what this looks like.
Can I ask a question here?
Yes, please.
So how does Nmax scale with the total number of resources in the system?
Yes, that's a really good question. So here in this particular example, we had just one resource that we were providing, and we saw that n max depends on sort of all of these parameters. So if you tell me what the parameters are, I can tell you how many species in principle can survive on this one resource. And now if we again continue using the assumption that one species can consume one resource, then just, if you provide me R resources, then it will just be R times this.
What I'm trying to get at is, in the model that you've sketched here, the effective resources are C0, C1, C2, and further on, whatever you have.
Yeah.
So what sort of a partitioning of the total niche space or resource space does this result in?
Yeah, I think the partitioning. So, I mean, of course, in terms of all of the resources, all of the species are perfectly partitioning the resources. They're not competing for anything. But in terms of how the original sort of energy in this one resource was partitioned across the species, that's something that you can calculate. That's something that you can approximate by looking at the relative abundances of each of the species. So that's something that I'm going to show you in a slide.
Thanks. Just a last point to make sure I've got it. So is N max just equal to the number of C's in the system?
N max is equal to not the number of C0s in the system, but
number
the
of C's, number of C0, C1, C2, all taken together.
Correct. But the job of calculating how many C's there will be in principle is what we've done.
Right. Thank you.
Actually, this can be mapped to a branching network.
Yes. Yeah.
Right? And so the question is really asking about what point the branching will.
Yes, correct. So you can sort of have this sort of picture where essentially, you have a microbial species which produces, more and more, and this network keeps branching. And at some point, this branching stops is essentially because species abundances are going lower and lower and lower. At some point, they can't beat dilution.
I wonder if a similar thing can be thought of like a mechanical network as well. For example, lungs branching.
Can a similar thing be thought of in a mechanical net? Thank you. In a mechanical network. That's an interesting question. And what would you think the components of this network, like what would the mapping?
Some fluxes of fluid flowing past.
Okay. Yes. Certainly. The only sort of constraint there that would need to be that stops this process at a certain point is that you need a replicating agent. That's sort of washed away for some reason, or has to pay some sort of maintenance cost.
Right.
So if you have something like that, then this kind of
but the washing away can happen everywhere throughout, right? It's not that it's only the bottommost layer.
Correct. It's happening everywhere, but the bottommost layer is the bottommost layer because that's the point at which dilution really gets you. Okay.
Hi, Akshit, could I ask? One is, I remember there was a paper by Ned Wingreen around the same time, right, where they were finding solutions where you could actually get unlimited number of species to coexist. And I just wanted you to comment on what limit they were in, or whether you can think of that in terms of your model. Was it just zero dilution? And second, this power law, could you say if this is because of the structure that you've built into your model, this kind of hierarchical structure, or you think it's more general than that?
Yeah. Okay. So the first question, which was that there was a paper from another group of Ned Wingreen, where they have a degenerate case where you can have, in principle, an infinite number of species coexist. That was sort of in the opposite limit of the calculation that we've done here, which is that here we have species using resources and producing byproducts, and that's what allows diversity. There they had, in principle, R different resources. Everyone could use every resource, sort of much like the sort of simplex model that I was describing yesterday. And there's a degeneracy, which is that all of the species have equal fitness. That is, all of the species are able, the sum of the consumption rates of their resources add up to the same. So that's a case where there's pure competition between species, and here's a case where there's pure cooperation between species. There's no competition between species except in those cases where they very transiently compete for the same resource. So I sort of think of them as think of these two results that came around the same time as these two different extremes. With regards to the second question, which was one about whether the power law structure comes from the hierarchical nature of the network that we've constructed, I think that's very much the case. As you saw in this calculation, basically, the reason that it came about was the fact that we had this sort of geometric series that tells us how many species can coexist, and that is fundamentally tied to the structure of the network that we imagine. Does that answer your questions?
Yeah, thanks.
Sorry, I just had one question. It's back to this the time scale of relaxation. Yeah. So you said that approximation is valid when delta is small. Is that what you said?
Delta is large.
Delta is large. I see. I see. Okay, so delta is large. So that means that this technique works. So Nmax is typically has to be small then, right? Because you have 1 over delta power something, right?
Yeah. So it's roughly of the order about 100 or so. The number of species it can survive.
I see. I see. So this. So if there are too many species that survive, then you cannot presumably use this approximation. Then probably, So how do you? So
correct. So yeah. So in principle, if you have a very, very low dilution rate, then you could have many species survive because you can get much lower down in this network, and in that case, things do start breaking down. And I can sort of show you that here on the left of this plot that I'm showing. Is the number of species that survive in the ecosystem as a function of the dilution rate delta, and the black line shows sort of this power law that we had with some alphas and beta plugged in, and the red shows you sort of results from simulations, and you can see a sort of this sort of starts diverging the lower and lower you make your deltas.
Oh, okay. Thanks.
That's correct. Yeah, correct. So we sort of imagine. So there's a slight complication in this, which is that we need to get the lambdas and so on and so forth, which here we assume were all some large common number lambda. In the simulations, what we do is that we actually simulate the random assembly of this ecosystem. That is, we have a pool of species with some random lambdas that we create. And we now start populating this ecosystem by throwing one species at a time, letting it equilibrate, and sort of building the ecosystem until it will, and sort of simulating it for some long time and seeing how many species we get. And so we do it many times. This process, this sort of random process, many times, and that's what gives us some number of species.
So you're saying your results are actually a good answer even when the lambdas are not the same.
That's correct. Yes.
That's what this is showing.
Yeah, and so essentially the reason that is true is that in the even though the lambdas are not the same, you get many species with different lambdas populate the ecosystem in the middle in sort of the initial transient, but sort of over a long time of ecosystem assembly, they all get replaced by sort of the best species consuming that resource. So even though you have some, whatever distribution of lambdas that you assume, you end up at some extreme value of lambda in all of these.
And if you did the more general case that Loga was discussing earlier, where there was some competition and some cooperation and they were all consuming everything, then would this still, that you could easily simulate numerically.
Correct.
Would this still be a good answer for that?
It actually doesn't seem to be a good answer. And so some of that is because when you're doing that simulation, there's not one way that you can do it. You have to make many different model choices. And there's some choices you can make where it continues to be very good, and then some others that you can make where sort of things get pretty bad, and we can talk about that later. Yes.
Yeah, I was just wondering whether this geometric series or this what is at the bottom of it, the fact that the number of resources grows exponentially in some sense.
Correct.
The beta is
the lower and lower the layers get, right? Yeah.
How? That sounds like a very strong and very too strong an assumption, because you would think, okay, it's taking in the resource C0, and there should be a limiting factor that C0 might break up into C1, C2, and then maybe C1 breaks up. But that layering is probably going to stop much earlier than the species layering, no?
Yeah, correct. So in fact, initially when we wrote this paper, this is exactly the criticism that we got, which is that, you can what is this C0 going to be? You can think of it as being some sort of 6 carbon molecule, and you can break it down, from 6 to 5 to 4 to 3, but at some point it will stop. But and actually we agreed with them at that point, but I think over the last few years, what has become clear is that these C0 actually are not small carbon sugars, but they're essentially polymers with many different carbon bonds. So the number of times that you can actually break it up biologically is actually much larger than the Lmax that you typically get in these systems in the first place. So that doesn't seem to be a limiting factor, even though you're right, it certainly could.
So the Lmax, you're saying it would be of order, that's what...
4 to 6 or something like that, right? Okay. Yeah.
Not, the upper end, it was going at 100 or so.
Yeah, so this is about 100 species, and this is with beta about 2. So in this sort of egregious case, of course, you can get many. But yes, that would only be possible for really big polymers, which certainly microbes are capable of making communities on polymers, like starches and cellulose that we eat.
What is the kind of alphas that you're thinking of here?
The alphas that we're thinking of are roughly like 0.9 or so, in fact. So an alpha of 0.9 implies that basically 90% of what you eat is being thrown out as byproducts.
So very few it is actually going into the bacteria.
Very few is actually going into the bacterium. And again, at the time when we assumed this was just something that we assumed ad hoc, but I think over the past few years, people have actually made quantitative calculations of what this efficiency in various bacteria is, and it is rather close to 10 to 20%. So an alpha of about 0.1 or 0.2 is, 0.8 or 0.9.
Why are the processing efficiencies so bad? Is there some biological understanding of?
Yeah, so there's some understanding which has to do with the fact that there is usually a trade-off between rate and yield. That is, most of these organisms want to grow really fast, and as a result of some biochemical trade-offs, they have to suffer a very low yield. And so, that's for sort of chemical reasons that these yields are quite low.
Question here. If instead of this sort of model, you took a different approach and tried to look at it from the point of view of a neutral model, the way the phrase is used, what sort of abundance distribution would you predict?
Yeah, that's a good question. So certainly people do these things which are called neutral models, where you don't assume that species are different from each other at all. Rather, there's just migration processes and so on and so forth that decide the abundances in time. And indeed, they find that when they do those calculations, you still get abundance distributions, which are our law. But their slopes are quite different from the slopes that we see in these ecosystems, and that we would kind of expect from the biochemistry of how many byproducts they produce. And second, the diversity that you get in these neutral models is in principle quite lower than what you can get from these kinds of models.
Why would that necessarily be low? I don't get it. In both.
So you're right. It would be controlled by the migration rates and the death rates in principle. But again, when I say low, I mean low given the kinds of rates that we think are reasonable for these ecosystems.
Thanks.
I have a question.
Yes.
So in the bioreactor, we assume that the mixing is very well, right?
Yes. It's a fully well mixed system.
So if the mixing was poor, then the number of species could easily be several times the number of resources.
If there was no mixing.
Yeah, very poor mixing.
Correct. So if there was spatial structure, certainly you could get many more species.
So could you tell me, the mixing rate must be faster than some time scale in the system. So what's the time scale? Is it the reproduction rate or the time scale of evolution? So what's the time scale should we that we should?
I think one reasonable time scale that you might think of is sort of the time scale of clumping or settling of these different bacteria, because the way they sort of, what you do when you're mixing these systems is that you're sort of trying to fight aggregation or clumping that occurs. And so that happens at a certain rate. And so you just need to mix faster than that.
Right? So the system I have in mind is like, if I have a lake in which I want to see the number of bacteria, then and like depending on what the state of the lake, the mixing can be very different. So the rate at the if we can like, if I have some mixing, like I have some very mixed chambers and I have some, let's say, some coupling between them, then if we can understand the number of species as a function of the mixing rate, that might also be better for larger ecosystems.
Right. Yeah. So in principle, you could also like write down a model where there's some mixing rate. Yeah, but that's that's also something that one could do. Yeah.
My question earlier was on the right hand side. Is that actual data, because it says human oral microbiome?
Right. So on the right hand side, so just to go through this on the on the right hand side, I'm showing you the abundance distributions of various species, and in particular, I'm showing you rank abundance curves, and the solid lines show you sort of the abundances that we get from our model, and the dots actually show samples from a data set, where the red shows you a sample of the human oral microbiome, and the blue dots show you a sample of a bioreactor, which was sampled. So the dots are sort of blobs of the data, the lines are the model.
I see. Sorry. So the red dots actually are they matching the line very well, the solid line very well?
So so the red dot, certainly when the rank is lower, the agreement is not perfect, but as you go sort of down, and this would sort of correspond to the lower layers of the bacteria in our model, the agreement is very well, I would say.
I see.
How many parameters are there?
So here there are essentially two parameters. There's alpha and there's beta. And so the way that we get different slopes even is just by varying alpha and beta. For this particular thing, we didn't vary alpha at all because we think that the yield is something that's a rather universal parameter. The number of byproducts, beta, is something that could vary based on the chemistry of the system.
So the main result really is that the normalized species abundance and the species rank, they follow this slope, right?
Yes.
There's some logarithmic relation.
Correct.
I see. And is that a surprising result, or is that something you could have guessed from other ways, or is that something that was known earlier?
So
you would in general have this relationship. I mean, as you said.
Correct. So in principle, power law abundance distributions are something from observations that we knew of before. But again, the field is searching for models that can sort of describe these power law abundance distributions along with a bunch of other things. For instance, the number of species that can survive.
I see.
But can I not attribute this to the fact that you have two exponential? I mean, basically it's a competition of two exponentials just because you have a tree-like graph?
Yeah, no, that's absolutely correct. That's how you get it in the model. But I think what the model is trying to say is that this is actually how it might be happening in biology as well. And certainly, again, over the past few years, what we've seen is that these kinds of branch structures that the model assumed actually are quite a reasonable approximation. The way that we find it is that biochemists are actually able to trace these networks in systems over time, and they look quite like these branching structures.
Okay, and a purely academic question. So how robust are the predictions of your model if I relax the assumption that that a species BI can only eat the byproduct of just eat one byproduct of BI minus 1? So in other words, if the tree-like graph has your dynamical process has some links which sort of have longish loops.
Are you meaning to ask a case where a new species could eat more than one resource?
Yeah, for example.
Yeah. So I don't know how robust our results would be to that. Probably not very. Right. Correct.
Sorry, you probably mentioned this when these solid lines. When you say it's from numerics, you are solving the differential equations numerically with no further approximations. Is that correct?
That's Right. What we are doing is, first of all, we're simulating the assembly of the ecosystem over time. Yeah, we're not using any approximations for the lambdas being the same and so on and so forth. We assume different species with different lambdas and so on and so forth.
And then you extract the large time values, is it?
Yes.
I see. Okay. Thanks.
Can I ask a quick question?
Yes.
I mean, so is this, I mean, would you call this, is this a model for then competitive exclusion, or is it more, I mean, it seems like it's inclusion and cooperation, right?
Right. So the idea that we started off with was that if you had just one resource and no cooperation at all, there is no byproducts being produced at all, you could get only one species surviving. But then we learned that if you allow more byproducts to be produced, then in principle, you can break competitive exclusion, where you have only one resource that's being provided from the environment. Everything else is being created by the bacteria themselves, and they can then have many species survive. Right, yeah. Okay, so maybe I will...
just a minor question. For your numerics, how do you choose initial conditions?
You start off with some random small population size.
And the equilibrium.
Correct. Yeah. So we are guaranteed to have the same equilibrium, just one stable equilibrium in this condition. Okay. So so I will.
Sorry, just you should probably go ahead. But like the alphas and betas that you put in this model, you understand that matches with what you would know from biochemistry. Is that
correct? Right? Yes.
So you know the alphas and betas from the biochemistry of, let's say, the human oral biome.
That's correct. Again, we didn't know it at the time, but now we do know, and they match quite.
And they match with the parameters that you get by fitting.
Correct.
Okay. In how many systems? I mean, this is. I mean, you can look at a lake. You can look at someone's tongue, like you said yesterday.
Yes.
Someone's gut, or.
Correct.
I mean, how universal is it? This power law dependence.
The power law abundance distribution seem to be quite universal. The knowledge of what the betas should be in each environment, that's not, we don't know it so well for all of these systems, and so.
But you're saying, like in Loga's question, for the systems
that we do know it for, it matches quite well what the when you try to fit it with the data.
So there are a handful of such systems, maybe.
Correct. At the moment, there are a handful of such systems, about of the order of 5 or 10, I would say. But this is, of course, growing. Okay, so I want to shift gears a little bit. Yes.
How did you choose the lambda I values?
We chose them. Yeah, so we chose them randomly from a uniform distribution.
So doesn't that affect the final abundance of each bacteria?
No, in fact, it doesn't affect the final abundance of each bacteria, because the lambda only enters. Sorry, I raised it, but the lambda only enters as a small prefactor with delta square, and delta is already quite a small number. So the lambda actually doesn't affect the bacterial biomass so much.
Okay, thank you.
It's only alpha and beta.
That's correct.
Only two parameters you need.
That's correct, and delta to some extent, but the delta is sort of usually well known and well controlled in these systems.
Right.
Okay. Thanks. So I want to take my last 15 minutes or so just to change gears and talk about this other story that I brought up, which is one about multiple stable states. And maybe I'll sort of zoom past that, and then we can have more questions later if you want. I meant to do some proofs of that before, but I don't think I'm going to have the time, but please feel free to bug me if you're interested in that. So the sort of second story that I want to talk about was based on this idea that there are many other systems where we see microbes being quite competitive for nutrients. And so here's just an example from data where we have different microbial species at the bottom and sort of different nutrients at the top. And if there's a link from a species to a nutrient, and then that means that particular species can use that nutrient. And you can see that sort of there's large overlap in these networks. what we want to be able to do is that we want to be able to explain this phenomenon of multi-stability in these systems using this idea. Just in brief, the phenomenon of multi-stability is one where you have all of these species being able to survive, but in different stable states, they're able to survive at different abundances, right? And so this was a sort of question that was plaguing the field at the time that we did this, at which we did this work. People didn't quite know a good model to get these kinds of stable states. And so the idea that we came about to sort of explain this was to draw a sort of analogy between the stable marriage problem and microbial communities. And I'm going to sketch the idea in sort of the next 5 or 10 minutes. The idea is as follows. The stable marriage problem says that there are n men and n women that all, want to get married to each other. This is, I'm sorry for the anachronism. Please bear with me. They have some set of preference rankings. Every man has every woman in some rank in his preference list, and every woman has the same. And the job is to find a set of matchings between these men and women that is stable. And what is the notion of stability? The notion of stability is that to have no eloping or no blocking pair. What this just means is that in any matching that you get, for it to be stable, you should have it such that no man that is matched to a woman in a pair should prefer a woman that they're not matched to, and vice versa. That woman prefer that man more than her current partner. If you have any of those, you have blocking pairs, and that sort of matching is unstable. What sort of Goyal and Shapley showed in the 60s was that given n men and n women and any set of preference rankings, you are guaranteed to have at least one stable matching. And in fact, they gave an algorithm which is now called the Goyal-Shapley algorithm to find this stable matching in polynomial time, right? And so I just want to draw the analogy for you first, and then we can get to the results of multiple stable states. How does this all map to microbial communities and resources? Well, the idea is that instead of men and women in this problem of the stable marriage problem, you can think of microbes and nutrients as being the two agents, with the idea being that microbes have what are called nutrient preferences. As I was answering to a question that was asked previously, there are many cases in which microbes, when they're given sort of many nutrients to choose from, let's say this microbe M1, it's given nutrients N1, N2, and N3, it has an inherent preference hierarchy, which is given by some regulatory network, where there's a nutrient N1, let's say that it prefers the most, N2 the second, and N3 the third. And a different microbe N2, in principle, could prefer different sets of nutrients. And so there's some sense in which these microbes have different nutrients, and experimentalists can actually do experiments and measure what these preference hierarchies are. And at the same time, you might say, okay, fine, microbes have preferences and choices. What about nutrients? They're passive. And here we use the idea of competitive exclusion that we talked about earlier, which is that if there are any two microbes that are now trying to compete on a single nutrient, let's say N1, one of them, one with, let's say, the higher lambda in this previous model, will sort of win the competition for that nutrient. And so in that sense, the nutrients don't have a preference, but the competitive abilities of the microbes sort of set up a hierarchy of microbes, of one for every nutrient. And so in this way, you end up with sort of two ranked tables, one which tell you the nutrient preferences of each of the microbes, sort of what I've shown on the left, and one which tells you the competitive abilities of each of these microbes. And just by knowing these two ranked tables, which are somewhat easier to compute experimentally compared to knowing all of the various kinetic parameters, we can actually get the full stability landscape of these microbiomes. Not only can we get multiple stable states, we can calculate out exactly how many there are. In fact, we can show that in principle with n microbes and n nutrients, you get of the order n log n stable states. Right, and the calculation, that is something that I'm not going to go through now, but I want to show you some sort of easy cartoon examples that help sort of explain what kinds of new biology we can we can understand by doing this kind of mapping. The first is the fact that you can have communities which have a set of species surviving. You can introduce a transient invader in them. This is an invader in purple here that won't eventually survive. It will eventually go extinct, but at the end, it will have changed the state of the community. So, so let me just take you through that example here. Here we have these two microbes, the red and the yellow. And there's two nutrients N1 and N2. There's some preference. There's some preference tables that we have. We can have a new microbe, this purple microbe invade, and it, let's say, prefers nutrient N2 the most. It'll go and sort of try and compete for it. In the process, it will knock out the less competitive microbe, the yellow one out, which will now start to compete, and so on and so forth. This restructuring will go on until the purple microbe is eventually unsuccessful at establishing in the community. But at the end, you'll see that the stable state is reversed. This is sort of the second stable state of this system. The way that this maps to biology is that this is actually quite similar, we think, to how probiotics behave in our guts. There's sort of a lot of questions about the fact that we consume probiotics and they contain sort of a small amount of microbes, and we don't even often see these microbes that are in our probiotics survive after we've consumed them. And so people used to think in the past, how can these probiotics possibly change our gut and bring it to a healthy state, whereas they don't survive? Well, sort of what we're claiming here is that at least now with this mapping, we learned that it is indeed possible to find cases where you have a transient invader, which nevertheless doesn't survive, but changes the state of the community permanently. There's another example, which maybe I won't go through now in the interest of time in detail, which is that you can, instead of introducing a transient invader species, you can introduce a transient nutrient, which again, through this process can change the state of the community. But in principle, what you get from sort of doing this sort of analysis is that you end up being able to compute all of the stable states that the system can be in, given these ranked preference tables. So here's an example that I've drawn here. This is a sort of random set of preferences that I drew out on the left. And you can use the Gale-Shapley algorithm and sort of another algorithm to compute all of the stable states. The sort of interesting structure that you find in these stable states is that they obey a very particular hierarchy, which is that if you want to transition from one stable state to the other, you can do it by means of a very specific perturbation. What perturbation is that? That in this case happens to be transiently removing a particular nutrient and then reintroducing it later. So just to show you what that looks like in detail.
Sorry, could you explain this table?
Can I explain the table?
Yeah. If you can.
Right. So, yeah. So the table is one where the table at the top tells us for each microbe, this is sort of a different species in a different color, what its most preferred nutrient is, that's sort of rank 1, what its next preferred nutrient is, that is rank 2, and so on and so forth. And there's sort of this row, you have each row for each microbe. At the bottom, you have the competitive abilities of each of the microbes, that's in terms of ranks. So again, each of the rows are different nutrients. And again, you have which species is the most competitive on this nutrient, that's given in red, which species is the second most competitive, and so on and so forth.
So this is the data that you're...
Correct. Okay. Correct. So this is usually what you get from experiments.
So you just have an ordered thing. You don't have a numbers for each of them.
Right. Because usually what you do is that you put two species in competition with each other and you learn which one survives.
Okay.
And so you have sets of rankings.
And the statement is that, so given this data, you can set up a transition network on which state.
Correct. You can calculate exactly, given these sort of 7 species here, you could in principle have 7 factorial different states that you could have. But in this case, it so happens that only 5 of them are stable in the way that we describe, which is that no microbe would want to use another nutrient and be more competitive on that nutrient.
Okay.
Sorry, question about how the data is gathered.
Yes.
Are you saying that pairwise competition experiments are carried out?
Yeah. So for the bottom table, which gives you the competitive abilities, you can do pairwise competition experiments. People also do experiments with more than pairs, but just in a single nutrient environment to know which sort of species survives. At the top, what people do to know what the preferences of each microbe is that they grow each microbe in isolation with a whole bunch of different resources. They track all of their concentrations and they see which one is depleted first, which one is depleted second, and so on and so forth.
But if you depend on pairwise competition, you are assuming that these rank preferences are transitive, aren't you?
Correct. And so it often turns out that they are transitive. And this is an experimental result that's been shown a few years ago, that often these competitive networks are transitive.
But as far as I am aware, in many cases, they're not transitive. And that, in fact, is a major complication.
Yeah, certainly in some cases, they might not be transitive, and there our theory would not apply.
So
right.
What I'm getting at is the presence of microbe 3 might affect the relative preferences of microbes 1 and 2.
Correct.
And conversely, the presence of nutrient 3 and so on.
Correct. So these kinds of what are called higher order interactions are certainly possible, and people have indeed found them. But statistically, I would say from the data that I've seen, they happen to be rare. But nevertheless, when they are quite common and in systems in which they might be quite common, then our assumptions would break down in this, and then this theory would no longer. Okay, so I don't want to take up too much time, so I won't go into the details of exactly how we prove that this transition network is strictly hierarchical and how we prove that there are this many stable states. But in principle, what I did want to show you is how you go from one stable state to the other. Sort of one of the predictions that this mapping makes is that we can exactly predict how to go from one stable state to the next. And we do that by means of what is called a transient perturbation. Say that I have a certain stable state S2, where just to tell you how to read this stable state, in this stable state, the green species is using nutrient N1, the purple species is using nutrient N2, the sort of cyan species is using N3, and so on and so forth. We can imagine a perturbation in which we suddenly remove nutrient N1. What that does is that it can orphan the species that was using it, which happens to be the green species here. It will then go and try to compete on its next preferred nutrient, which in this case happens to be N7. And if it's more competitive than the current resident on this nutrient, then it will actually displace it. And so in this particular drawing of preference list, that's exactly what happened. That will make the red microbe sort of go to the next nutrient in its preference list, which in this case happens to be N1. And in this at this point, if you reintroduce nutrient N1, you will form a new state, which happens to be stable. And just to see that it's stable, all that we need to ensure is that this red microbe, which prefers nutrient N7 to its current partner, cannot actually go and recapture nutrient N7. The reason being that the green microbe is actually more competitive on it. And so by means of what is called a rotation algorithm, we can sort of introduce all of these specific perturbations and find which exact ones are capable of transitioning from one stable state to the next. And what we find is there are essentially two kinds of mirror networks. One that come from perturbing nutrients, that's sort of the one on the left, where every time you perturb a nutrient, you go to a different stable state, but you only go sort of down in this hierarchy. And sort of another mirror network where you can perturb the microbes, whereas you can transiently remove a microbe, have another sort of microbe come in, and that goes the other way, still hierarchically. And so in this sense, you might transition from any of the stable states that you have to any other.
So the way you've drawn it,
all the stable states are path connected in this graph.
Yes.
Is that always the case?
That is usually the case.
Is it always the case?
It's not always the case, but it's definitely true that this the organization of all the steady states is stable states is hierarchical. That is to say that you might have 10 or 20 different stable states, and so you might have at each layer many stable states, and so that's the sense in which it won't be sparse.
But from any given stable state, there is a way for me to.
Yes, there's only one in and one out link.
No, but suppose I want to go from state S1 to S10 or whatever.
Yeah.
There is a path of perturbing nutrients in such a way that I can start at any state and end at any state. That's what I mean by path connected.
Sorry, path. Yes.
If you've got a graph,
yes,
I can get from any vertex to any other vertex through some path.
Correct. Yes. Yes.
And I want to know, are all the stable states?
Yes, there are no isolated stable states that you can't get to by means of this perturbation. And in fact,
is that a theorem?
Yes. There's a theorem that proves that all of the states are connected by means of these perturbations. All of the stable states are connected.
Sorry, I didn't understand that. How can you, in the left graph, how can you get from S4 to S1? I thought it's a directed graph.
If you were just restricted to perturbing nutrients or perturbing microbes, you couldn't do that.
I see.
Yeah. So if you allow yourself to perturb both, you can go from any to any. But of course, if you just allow yourself to perturb nutrients, then of course you can't go from S5 to S1.
And why do you have this on the one in? I mean, you said something about one in, one out, but that's not
with the self loops.
No, so you said something about one in and one out. So I mean, okay, the question is that from S1, what happens if I perturb nutrient N1?
Right. Okay. That's a good question. So the self loops are sort of trying to show that if you try to perturb any of the other nutrients apart from N5 and N7 in this case, you will actually get back to the same stable state that you started. Okay. So it's 5:10. So I won't. So I won't go deeper into this, but sort of. That's what I have for you. And if you're interested in sort of the more mathematical details of how we get the number of stable states and how we can prove that this network is the way it is, then please feel free to bug me at any time. Are there any more questions? Thank you.
Questions, comments? I had a general question. So all of these are modeled as networks. So where's the microbiome here? I mean, it could be any network.
Right. So the microbiome itself, the community in this sense is can be in one of these states, right? So in each state, what's happening is that you have these various microbes present and they're using different nutrients, and as a consequence of using different nutrients, they'll have different abundances, right? If I use nutrient N1 versus nutrient N6, I might grow differently on it and I might have a different abundance. Maybe I don't understand your question perfectly.
What is the aspect of the ecology which is there in these graphs? I mean, it could be some gene interaction networks, protein interaction networks.
Sorry, I can't fully hear you.
It could be some gene interaction networks, protein interaction networks, whatever it be. What's the ecology aspect which is there in these graphs?
Right. So the ecology aspect that's there in these graphs is basically exactly how you transition from one state to the other. The way that you do that is by perturbing, perturbing the way that competition is occurring between different species. So in this example that I showed you here, what you do is you have a certain state in which some species are using some nutrients. By transiently removing a nutrient, you're sort of allowing a new set of competitive restructuring to occur, and that sort of competitive restructuring is where the ecology happens, and so that's defined by means of which species use which resources in which way. So the
relative strengths, I suppose.
And and here you don't need the relative strengths. Here the ranks are enough to sort of tell you. Just because I only need to ask when this green species goes to nutrient N7, I just need to ask if I wait a long enough time. Correct. So I just look at the table whether green is more competitive than red, and if it is, then I know that eventually if I wait long enough, it will replace it. And so that's really the ecology of which species wins out the competition on which nutrient.
The questions, comments, people online?
So I had one question.
Yes, I'd like to ask a question.
Please.
Sanjay?
Yeah, right. I just wanted to ask that these two parts of your talk, your last part suggested a kind of network of interactions that exists between species, between the bacterial species in ecosystems, in these microbiomes. And the structure was that every nutrient was actually connecting two species. It was being emitted by one and being ingested by another.
Right.
I mean, so if that is the kind of structure that of interactions, or the basis of interactions that that microbes have in microbiomes, how does it gel with this picture here, wherein where you have effectively those two matrices that you connect, those two ranked lists that you connect, have another kind of a network structure, it appears, right, which is not the same as the one that you talked about in the previous one.
Yeah.
Or are there some things that you can reconcile the two, or what? I mean, is there something that you've thought about this?
Yeah, no, that's a nice question. So so I would say there are two things that I would say to this. The first sort of reconciliation that I have is that even though the network that I showed you here was one where every species was in principle capable of using every nutrient, it was actually only ending up using one nutrient in one of the stable states. That is, it had a preference hierarchy, and in each stable state the instantiation of each species was one where it was using only one nutrient. So you might imagine, in sort of the first part of my talk, you might imagine that these species may in principle be able to use many different nutrients, but given an environment with many different nutrients, they start by using one, the one that they prefer the most and are most competitive on. The second thing that I would say to this is that, certainly these are two different sets of assumptions, and perhaps they apply to two different kinds of systems. The sort of second picture that I described to you, where there's a lot of overlap between microbes and the nutrients that they consume, might be one that might apply to our gut, where there's a lot of different nutrients that we get from our diet. Whereas sort of the first kind might apply to different kinds of ecosystems where the environment in principle supplies a very small number of nutrients, and then the further large number of nutrients are created by the microbes themselves. And so these are essentially two different models that would apply to two different kinds of environments.
So in the second case, is there some something that you can calculate from the model that you can compare with experiment with data?
So certainly one thing that you can calculate is how many so given a certain number of species and random sets of preferences, how many stable states you should observe. And typically what we see is that for systems with about 100 or so species, you observe about between 5 or 10 stable states in our model. And that's something that we can compare with data, because we have data of lots of different sort of stable state snapshots of various ecosystems like our gut. And there again, we see the number of stable states being roughly of that order. That is one. The second thing that we can do is that we can actually take experimental cases where people have data about these tables, of these kinds of tables, and then predict exactly which perturbations should be performed to be able to transition from one state to the next. And so that's also something that that one can compare with experiments.
Okay, thanks.
Was another question online?
Yeah, so my question was somewhat related to Sanjay's, and he's asked it extremely well. So I just had one more addition about how maybe you have thought of reconciling these two models, which is that, like you said, maybe we say N1 is equivalent of C0, and N2, C1, N3, C2, and so on. And you have these bacteria themselves also have the hierarchy where one is feeding off the produce of the other, as well as you have this preference list, so they can probably move up and down in this in the hierarchical model you initially described, the first one.
Correct.
Does that make any sense? And have you thought about that?
Yeah. I mean, so certainly you could think of it like that. The only sort of thing that then you will have to think about is what triggers a species to sort of change what preference it should use. One way that I sort of showed you here was if you have a transient removal of one of the nutrients that it's using, but certainly there could be other ways as well. For instance, if the concentration of that nutrient becomes quite low.
Right. Again, sort of something like this competitive exclusion you have in the first model itself. So you introduce a foreign, an alien species that has a lambda I that wants to consume N2 at first preference and has a lambda I higher than, say, the species currently consuming. So it displaces it, and then this one again wanders around, finds that it consumes N3 better than the green species.
Right. Yeah, correct.
So have you? I mean, does it make sense to even think about such a model?
I think it would, though. Again, like this is not something that we have followed up on. But yes, certainly, I think that you could think about a case where in this model, the different species are basically going and trying to use their first preference, and if they're unsuccessful, they go and try to use their second, and so on and so forth.
Right.
But that's not something that I've tried to solve yet.
Thank you.
Questions, comments?
So, continuing on this theme of reconciling, like how does these two things that you talked about today connect with what you're talking about yesterday about the strains and functionality?
Right.
Is that something that you would like to comment on?
Yeah. So like I was saying in the beginning of my talk, there's sort of different phenomena, one which has to do with the dynamics of the system, one which has to do with how many species you have, and one which has to do with how many stable states that you have. And there's sort of three different models for three different phenomena, but sort of the spirit that they share is that using of resources and the way that different strains and species use resources is one that's important. So simply, instead of thinking about how close together two species are, one can think about how similar they are and how they consume resources. So in yesterday's talk, we were talking about how similar they were on this simplex of resource consumption rates. In the first part of today's talk, we were talking about which particular byproduct or resource they were capable of consuming. In this final part of the talk, we were talking about how similar their preferences or competitive abilities are. So in that sense, a functional description of what these species can do with resources is a tying thread to explaining all of these phenomena.
Okay. One another question. So you talked about this competitive restructuring and so on.
Right.
So just to know, like, is that something that is known in the stable marriage problem within game theoretical context, or?
Yes, in some contexts. So for the n equals n case, it's certainly known that you can have what are called rotations, but the sort of general case when you have n microbes and m nutrients in principle was something that was not known.
So that
and that also, I mean, so you can sort of extend those proofs and see that that's true.
Okay. Thank you.
Since we are meeting shortly after this, you can tell me if it's a long answer. You can tell me separately, but when you have multiple stable states like the ones that you mentioned over here, that's one statement about the sort of the stability of this thing, but it's usually in nature or in dynamical systems, a system which has multiple stable states, especially a very large number of stable states, you may not be able to go very easily to the
yes. They might not be accessible.
There are, it's a large landscape, and you have to find that stable state. It may not be
correct.
So shouldn't that constraint? I mean, what you've said is a kind of a statement about the absolute equilibrium in a given the same, but the approach to that equilibrium, the ability to even find that equilibrium, where could
so the ability to find that equilibrium is actually what I was trying to show on this transition network here, which is that one can prove for this particular formulation of the problem that all of the stable states are in principle connected on this network. And so
landscape, everything is connected.
Correct. And so not only that, we know how to transition from a state to the next because we know exactly which perturbations we can perform. So the question now only becomes about how you reach any one of these states. So if I can get to any one of these states, let's say I can get to S1, then I can get to all the states because I know exactly how to perturb this state to get to any of the others. The question of how to get to state 1 was one that was solved by was one that's given by the Gale-Shapley algorithm, which is that the algorithm says that given any n men and n women, this was the stable marriage formulation, there exists at least one stable state that happens to be the S1 that I've shown here, and that there exists a polynomial time algorithm to find it. And in the process of writing that algorithm, you get to that state.
Right, but nature follows that algorithm?
Whether or not nature follows that algorithm is a good question. So the algorithm is one that's couched in terms of microbes going and trying to use their first preference for, first and seeing if they can survive on it, then going and trying to use their second preference first. We have some limited evidence of this, which is that, for a low number of species and resources, people can actually track these kinds of dynamics. And there we know that it works reasonably well. But for large N, we don't have the data in particular to know whether something esoteric is happening there. Yeah.
How high dimensional spaces are you working with? No, because this question is very, very important. When as you know very well, I mean, it's a lot of that, how high dimensional spaces in the gut, for example?
About 100.
Oh, that's all.
Right. Yeah.
That's not a very high dimensional space.
Yeah, but, and you're saying...
yeah, I don't know what
Right.
Where this problem with Rajesh is alluding to happens.
Right.
It doesn't happen in 100 dimensions. I know that.
It's 100 species.
Right. Yeah. So that's
100 factorial.
This is huge.
Correct. So in principle, So in principle, here the total possible number of stable states is n factorial.
Right.
The point is that the number of them that are stable actually is much smaller, roughly n log n or something like that.
Okay. N log n. Okay. Yeah. So that's about a 10 or 15 or something.
Yes. And that's indeed what we observe. Thank you.
Thanks, Akshit. Thanks everyone for joining online. See you soon.